So let's talk about a technology that is, and I mean this literally, rewriting the code of life itself. It's a discovery that promises to cure some of our most devastating diseases, but it also opens a Pandora's box of questions we are just not ready for. We're standing at a crossroads, and we need to figure out which way to go. Think about it like this. The instruction manual for a human being is about 3 billion letters long. Now, what if you could find one tiny single typo in that massive book, a typo that causes something like sickle cell anemia, and just fix it, like hitting backspace and typing the right letter? That is the incredible promise of CRISPR. But right away, this incredible power splits into two very, very different paths. On one side, you have the power to cure, to literally erase diseases that have haunted us for centuries. But on the other, you have a path towards engineering humanity itself, creating what people are calling designer babies, with traits that we pick and choose. And that, right there, is the heart of the whole debate. And this is the best way to think about it, and it's an idea we're going to come back to. Think of CRISPR as the sharpest kitchen knife humanity has ever held. In the hands of a masterful chef, it could cure hunger and end untold suffering. But in reckless hands, it could poison generations. Okay, so how does this amazing tool actually work? Well, it's not some brute force weapon. It's way more elegant. Think of it like a pair of hyper-precise, GPS-guided scissors. And the origin story here is just wild. Scientists found CRISPR back in 1987, but not in some fancy government lab. They found it in yogurt bacteria. It's an ancient immune system that bacteria use to fight off viruses. It's like a tiny heist story. The bacteria grabs a piece of the virus's DNA, like a mugshot, and if that virus ever shows up again, bam, it knows exactly what to look for and how to chop it up. It's a brilliant natural defense that we've now figured out how to use. And it really boils down to 3 simple steps. First, you have something called a guide RNA. Think of it like a GPS coordinate you plug in. It takes an enzyme called Cas9 directly to the faulty gene you want to fix. Second, Cas9 acts like molecular scissors and makes a clean cut, snipping the DNA right there. And finally, the cell's own natural repair crew comes in to fix the break. Now here's the magic trick. We can fool that repair crew into either just disabling the bad gene or pasting in a brand new, healthy one that we provide. Find, cut, repair. It's just that elegant. And this incredible science is already producing results that, honestly, you can only describe as miracles. 44. That's how many sickle cell anemia patients are now, for all intents and purposes, cured. They're living without needing blood transfusions. One of them is a teenager who, after 5 agonizing years of constant pain, can finally just live a normal life. This isn't science fiction. This is real, right now. And it doesn't stop there. Take a rare form of inherited blindness. A single CRISPR injection, one shot, into the eye, restored functional vision in 79% of the eyes they treated. People who were going blind can now see. It's just incredible. And its power goes way beyond just human health. In some trial areas in Burkina Faso, scientists used CRISPR to basically edit mosquitoes to be sterile. And the result? A mind-blowing 99% drop in the local malaria-carrying mosquito population. Just think for a second about the millions and millions of lives that could save. Okay, but with every single one of these triumphs, the shadow of that sharp knife, it grows a little bit longer. So we need to slow down here. We have to. And really confront the profound, complex, ethical maze this technology creates. To really get why people are so worried, you have to look back. The idea of improving the human gene pool isn't new. The eugenics movement in the 1920s led to absolutely horrific things like forced sterilizations. Then, flash forward to 2018. A scientist in China, He Jiankui, did something the entire world had basically agreed not to do. He created the first gene-edited babies. That single act ripped open all these old wounds and lit a fire under the debate we're having today. And the story of those CRISPR babies is just a tragic, cautionary tale. He claimed he edited their genes to make them resistant to HIV, but the procedure was reckless. It created what are called off-target mutations, mistakes, in 5 other genes. Because of this experiment, 2 little girls, Lulu and Nana, might now live their entire lives with a higher risk of getting cancer. And this brings us to the question, the one we all have to think about. Curing a disease feels right. But should we use this incredible power for enhancement, to edit for intelligence, for height, for athletic skill? Where do we draw that line? Well, if you look at the data, it seems like most people have a gut feeling about this. There's pretty wide support, about 60%, for using CRISPR as a therapy to cure disease. But that support absolutely plummets, down to just 20% when you start talking about enhancement. There's this strong feeling that one is healing and the other is playing God.
And then there's the other huge question. Who gets this? Right now, 90% of all CRISPR clinical trials are happening in the US and Europe. At the same time, there are over 1 million people with the sickle cell gene in India alone who have almost no chance of getting this treatment. So is this amazing technology just going to make the gap between the world's haves and have-nots even wider? So where do we go from here? How do we wield humanity's sharpest knife without causing irreparable harm? Well, the future isn't just about wrestling with ethics, it's also about making the science itself better and safer. The version of CRISPR used to make those babies, it's already becoming outdated. New systems, like something called prime editing, are way more precise. Instead of making that risky cut through both strands of DNA, prime editing works more like the find and replace function in a word processor. It just swaps out single DNA letters. This reduces the risk of those dangerous off-target mistakes by over 100 times. The tools are getting so much safer.
And there is a real global effort to build some guardrails here. The World Health Organization has called for a complete stop to any editing that could be passed down through generations. In the US, the FDA is approving therapies for sick individuals, but it's banned editing embryos, and we're even using AI to design better, safer guides, while big funds are finally being raised to tackle that massive problem of access for everyone. Which brings us all the way back to where we started. That sharp kitchen knife. It holds the potential to create a feast of cures for everyone. But we have to move forward with the skill and the deep caution of a master chef, because one reckless move could literally poison the human gene pool for every generation that comes after us. The science is moving at an absolutely breathtaking speed. And that leaves us with a profound choice. By the end of this decade, are we going to look back and see CRISPR as the tool that saved us from our worst diseases, or the one that started creating a new engineered class of superhumans? The choice and the responsibility is ours.